All right, thank you again to our presenters for their fantastic presentations. We will now begin our question and answer panel. Joining our speakers for this panel is Dr. Peter Bross, Chief of the Oncology Branch of CBER's Office of Therapeutic Products. If you haven't had a chance to enter your question into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Now we'll start with a question that came in for Heather Erdman. Um, is it difficult to get an Interact meeting granted by the FDA?
That is a good question. It is not difficult to get an Interact meeting granted as long as the criteria of an Interact meeting is followed. The stage of development and the meeting package, format, and content, including how the direct questions are posed, have an impact on this decision. Using the direction provided in the Interactions with OTP webpage and the related guidance document in SOPP 8101.1 can provide that specific guidance that you can consider for your current need. Thank you.
Thank you so much for that response. Another question for you. Can a Type D meeting be requested as the first meeting with the agency? Sometimes there are simple questions that would be nice to be answered without the detailed background needed for an Interact or a PIND.
That's another great question. Yes, a Type D meeting may be considered for a first meeting, again, as long as the criteria of a Type D meeting is met, such as no more than 1 to 2 topics with no more than 3 to 5 questions posed, including a restricted number of disciplines. Please note that a limited number of Type D meetings may be granted in a close period of time, and these could be combined to make up one or another meeting type, for instance. Therefore, prospectively planning when and what meetings you want held with FDA aligned with your product development timeline really is ideal. Thank you.
Thank you so much for sharing that information. And now we'll move on to a question for Dr. Karin Knudson. How much stability data is required for cell and gene therapy IND submissions?
Great, thank you for this question, and it is a great question. So the amount of product stability data required for your product will of course depend on the phase of the clinical study. However, I do want to make the point that stability data is required in all phases under the IND to establish a shelf life for your product and to demonstrate that the product remains within acceptable limits for the duration of your clinical trial. So typically, we expect preliminary stability data, especially for the in-use product. We expect that to be available for the start of your phase 1 study. So you should submit preliminary stability data for your product in your IND, as well as your stability study design plan for long-term stability studies. Thank you.
Thanks so much for that response. We have another question for you, Dr. Knudson. When does the FDA expect potency assays to be developed and used for release testing during the product's life cycle?
So thank you for this important question. We recognize that potency assay development is generally difficult for cell and gene therapy products given their complexity and at times their inherent variability. So potency does apply at all stages of clinical development, but your potency assay is required to be used during the clinical studies intended to provide the primary evidence of effectiveness to support a marketing application. So this would be your pivotal study, and that is when that potency assay needs to absolutely be implemented. Your potency assays may not be completely defined early in product development because your product and process knowledge is limited, but it should become progressively more comprehensive as you accumulate manufacturing experience. Following up on what I said in my presentation, we highly recommend that you start product characterization and potency assay development early, even during your phase 1 study or during your preclinical studies, so you do not run into issues later during the product life cycle. Not having an adequate potency assay in place is an issue for our sponsors going into their pivotal study. So overall, we recommend that if you do not implement a potency assay for release testing for your early phase studies, you should provide in your IND submission a description of your general plans for product characterization and potency assay development and how you will reassess your potency assurance strategy during product development. Thank you.
Thank you so much for that thorough response. Now we'll have a question for Dr. Devaveena Dey. Dr. Dey, can you compare and contrast the differences in CBER's use of the NOEL or NOAEL versus a MABEL approach?
Hi, thank you for that question. Yes, that's a very interesting and important question. While I should speak to NOEL, which is more for cell and gene therapy, that is the no observed adverse effect level, which is typically the highest safe dose level for a cell and gene therapy product. However, a MABEL, which is traditionally used for monoclonal antibodies, is the minimal dose level at which one sees biological effect. So both look at pretty different aspects. However, I think the important thing is both look at. Thank you.
Thank you so much for that response. We have another question for you, Dr. Dey. When you mentioned GLP compliant toxicology studies conducted by a certified testing facility, does FDA certify the testing facility for a GLP laboratory organization? If not, what does it mean by a certified testing facility?
Thank you for that great question. To clarify, FDA does not certify GLP facilities. However, there are multiple and several GLP certification programs and organizations who do the certification, and those are all recognized by the FDA. Some of such certification programs are those issued by the CT, CFPI, and there are several other such programs. However, FDA does not certify any program. Thank you.
Thank you so much for that clarification. And now we have a question for Dr. Peter Bross. We did phase 1 studies in Europe about 10 years ago. We are planning to do phase 2 studies in the USA. Do we have to submit an IND or base our IND in Europe? Can we do phase 2 studies in the USA?
Okay, great question. Can you hear me okay? The answer is that you need to submit an IND to do a study in the US. And we see this more and more frequently that there are clinical development programs which are based on studies initially conducted in different countries, but not under a US IND. So in order to do the studies in the US, you will need a US IND. And if you have already performed clinical studies, I would recommend that you request a pre-IND to discuss, because there may be some differences in requirements for product and preclinical evaluation. And we're seeing more and more of these studies that are clinical development programs that are initiated internationally, and then they come to the US. And we have a specific guidance on that and a specific code of federal regulations. But my suggestion would be to come in for a pre-IND to discuss the requirements.
Thank you so much for that response, Dr. Bross. Another question for you. What is your recommendation on FDA notifications for studies to be conducted internationally? How early should those notifications occur?
Okay, well, this is kind of an interesting question related to the other question, but depends what you mean by notifications. As I mentioned, that in order to do a study in the US, you would need to submit an IND and have the IND be in effect. So the IND needs to be in effect before you do the studies. You can also do foreign clinical studies under a US IND, but it's really just required that you have an existing and open and in effect IND in order to initiate studies. And if you're studying, conducting studies internationally, then it's up to the sponsor when you want to notify us. But I think it's generally a good idea to request a pre-IND after you've had some clinical experience. Thank you.
Thank you again so much for that response. And now we'll have a question for Loni Henderson. If we've already met with CBER about our product, can we still call Matt B with a general question unrelated to our submission?
Thank you for that question. Yes, you can call Matt B with a general question so long as it does not have anything to do with your meeting with FDA or any other submission.
All right. Thank you so much for clarifying. We have another question for you, Loni. Sorry, just a moment. If we have an emergency and it is after close of business, whom should we contact?
Thank you also for that question. If you have an emergency, such as an emergency IND request, and it is after close of business, you should contact FDA Emergency Operations, and that information is available on FDA's website. Thank you.
Thanks so much for that response as well. And now we have a question for Dr. Jessica Lee. What is your general approach for grade 5 or death toxicities after receiving investigational cell and gene therapy products?
Thanks for the question. This is a great and important question. As pointed out in my presentation, the main purpose of early phase, including first in human cell and gene therapy study, is to demonstrate the safety of the investigational product. Therefore, we usually take every grade 5 toxicity, which is death, very seriously. In general, we will carefully review the product certificate of analysis and the clinical data, which will include eligibility criteria, the actual dose received, and the comprehensive medical record focused on baseline and post-treatment clinical information, and the autopsy result if available. Thereafter, based on the outcome of the safety assessment and the identified risk associated with the treatment, we will usually consider amending the protocol to revise trial design, eligibility criteria, dosing regimen, DLT criteria, stopping rules, safety monitor management, and supporting care to mitigate the risk associated with the investigational treatment. Thank you.
Thank you so much for that response, Dr. Lee. We have another question for you as well. Could you please discuss further on the long-term follow-up plan for CGT study?
Yeah, thanks for the question. Each cell and gene therapy are unique, and many require long-term follow-up. In general, the requirement and duration of long-term follow-up will depend on the product. And we usually adopt a risk-based approach to determine the requirement and duration of long-term follow-up. There are several factors taken into consideration, which include whether the manufacturing of the cell and gene therapy product involves gene editing or integrating a viral vector, whether there's a long-term expression of the vector or latency or persistency or reactivation. For example, we generally recommend up to 15 years for gene editing product and up to 15 years of integrating vector, such as retrovirals, lentiviral based and transposon element. And up to 5 years for AAV, which is non-integrating but with long-term expression. So in general, this will be based on the product and will be considered a case by case. Thank you.
Thank you again for your response, Dr. Lee. Now we'll address a question that came in for Dr. Karin Knudson again. Does the potency release assay need to be qualified? If yes, when?
Yes, I am happy to address this follow-up question. So yes, very short answer, yes, your potency assay will need to be qualified. The longer answer is that it will need to be qualified by the time you start pivotal studies. However, even if qualification of the assay isn't required, we generally recommend that you evaluate assay performance at all stages of development, because assays that don't measure what they're intended to measure, or assays with high variability or little robustness, will provide you with little to no information regarding meaningful control of your product. If you do have additional questions about potency assay development, qualification, or validation, I recommend you search for our potency guidances. There's actually a new draft guidance from last year, from 2023. I also recommend that you come in for a meeting with us if you run into issues with the development or the qualification or validation of your potency assay. Thank you.
Thank you so much for that response as well. We may have time for one more question. This question came in for Dr. Devaveena Dey. So GLP animal studies were conducted by another sponsor and will be cross-referenced by the new sponsor. Does that mean that a new sponsor is not eligible for the Interact because some GLP studies have been completed?
Hi, thank you for that question. In short, no, one can definitely apply for an Interact meeting, even though they do have some data which can be cross-referenced. So yes, for an Interact meeting, typically sponsors do not have data. However, if somebody can cross-reference data which applies to that particular program, you can certainly come in and apply for an Interact and refer to this existing data. So yes, you will absolutely be encouraged to do that. Thank you.
All right, thank you so much for that response, Dr. Dey. And looks like that is all the questions we have time for in this session. Thank you again to all our presenters and panelists for all the excellent information. Now we'll take another short 10-minute break and we'll resume at 1:50 Eastern Time.